A virus possesses no independent metabolic machinery. It cannot generate energy, and it cannot divide. It is strictly an obligate intracellular parasite. Instead of a living organism, a virus functions as a highly structured geometric delivery vehicle. Its sole purpose is to transport a strict genetic payload from one host to another. This microscopic image shows an icosahedral viral capsid. It's built with mathematical precision, forming a dense shell to protect the fragile genetic code inside. Other classifications take entirely different physical forms. This electron micrograph shows a highly complex, elongated structure, revealing how varied these non-living delivery systems can be. This diagram illustrates the bacteriophage T4, the baseline for viral architecture. The elongated icosahedral head contains the payload, a tightly coiled DNA strand. Below, a hollow tail and articulated fibers act as landing gear to lock on to target cells. The precise physical structure and the specific nucleic acid type of a virus represent a hard-coded algorithm. That algorithm dictates its exact strategy for hijacking a host cell. This diagram maps the lytic cycle. Tail fibers bind to a specific receptor on a bacterium. The outer sheath contracts, piercing the cell wall like a syringe. The phage injects its DNA directly into the cytoplasm, leaving the empty capsid outside. Once inside, the viral DNA executes its first directive, the immediate hydrolysis, or breakdown, of the host bacterium's own DNA. With the host genome neutralized, phage DNA uses the bacterium's raw materials to synthesize new viral nucleic acids and proteins. These assemble to form new phage components. Finally, massive accumulation of virions burst the cell wall, releasing new viruses. The lytic cycle is an immediate, aggressively destructive hijacking. It rapidly converts cellular machinery into a viral assembly line, destroying the factory upon completion. Destruction is not the only pathway. Some phages execute an alternative algorithm that prioritizes stealth and long-term propagation over an immediate kill. This diagram details the lysogenic cycle. Instead of destroying the host genome, phage DNA physically embeds within the bacterial DNA as a silent prophage. The bacterium reproduces normally, passively copying the prophage into datter cells until an environmental signal triggers the destructive lytic cycle. Lysogeny allows a single viral genome to multiply exponentially across a population, piggybacking on natural host division before executing its destructive code. When we shift from bacterial hosts to complex eukaryotic cells, the algorithms adapt. Enveloped viruses, such as HIV, bypass mechanical injection entirely. This diagram shows the eukaryotic entry pathway. Attachment relies on strict interactions between viral glycoprotein spikes and host receptors. Instead of piercing the cell, the virus triggers endocytosis. The plasma membrane invaginates, swallowing the virus into a vesicle before uncoating releases the nucleic acid. By utilizing endocytosis, the enveloped virus effectively weaponizes the eukaryotic cell's own ingestion mechanisms, tricking the host into pulling the invader inside. Once the genetic payload is uncoated, its exact biochemical makeup dictates its physical destination inside the cell. This schematic traces two distinct replication pathways. On the left, DNA viruses physically transport their genome into the host's nucleus to co-opt nuclear machinery for transcription. On the right, standard RNA viruses bypass the nucleus entirely, using their cytoplasmic RNA directly as a template. For these RNA viruses, every step, translation, genome replication, assembly, and release, happens strictly in the cytoplasm. The host's nuclear membrane serves as a definitive spatial boundary, strictly separating the replication strategies of DNA and RNA viruses. Biology rarely operates without exceptions. Retroviruses bridge the gap between RNA genomes and DNA integration, violating the central dogma of molecular biology. This flowchart maps a retrovirus. After uncoding, reverse transcriptase uses the viral RNA to synthesize a complementary DNA strand. This proviral DNA breaches the nuclear envelope and permanently integrates into the host's chromosome, forcing the nucleus to transcribe viral genes. By integrating into the genome, retroviruses permanently alter the host's genetic identity, turning the cell's own inheritance into a mechanism for viral propagation. Viral replication encompasses a broad range of evolutionary strategies, each dictated by precise structural geometry and specific genetic payloads. Regardless of the pathway, whether it's immediate lysis, silent integration, cytoplasmic replication, or nuclear hijacking, the objective remains mathematically constant. The virus must subvert host resources to build more viruses. This all culminates in the assembly stage, where newly synthesized capsids, fibers, and nucleic acids automatically snap together. For enveloped viruses, release operates differently than a bacterial burst. As the newly assembled virus pushes outward, it tears away a piece of the host's lipid viral layer to form its own outer envelope. During this budding process, the virus deliberately picks up fragments of the plasma membrane containing newly minted glycoprotein spikes, completely equipping it for the next infection. The viral life cycle functions as a hard-coded biological algorithm, converting a living, metabolizing cell into a production site for non-living code.